Hi everyone, welcome to Neurobiology at Providence College. I'm Joe DeGeorgis. Over the course of the next two lectures, we're going to talk about the discovery of the blood-brain barrier. And the first paper we're going to look at is by Tom Reese and Morris Karnovski, titled Fine Structural Localization of a Blood-Brain Barrier to Exogenous Peroxidase. It was published in the Journal of Cell Biology in 1967. But before we start talking about the paper, there's a little bit of background that we need to talk about. Prior to the work by Reese and Karnovski, a German physician named Paul Ehrlich did a very simple experiment. He took a mouse and he injected a dye into the mouse. And the dye flowed throughout the body of the mouse, with the exception of the brain. And he couldn't figure that out. Was there some intrinsic property of the brain tissue that stopped the dye from staining the brain? Or was there something inhibiting the dye from getting into the central nervous system? His student, Edwin Goldman, then decided to inject the dye straight into the brain and showed that the dye would indeed stain the brain tissue. So it suggested that there was some sort of barrier, and they termed this the blood-brain barrier. The blood-brain barrier is a very important structure. It protects the brain and it separates the circulatory system from the central nervous system. But it also complicates medicine because drugs that might be directed towards the brain, towards neurological diseases and disorders, can be blocked by the barrier. So if you inject the medication into your arm or you take orally some sort of medication, will that medication get across the barrier? The work by Ehrlich and Goldman also begs the question of what exactly the blood-brain barrier is. And that's the issue taken up by Reese and Karnovski in their publication. Again, the title of their publication is The Fine Structural Localization of a Blood-Brain Barrier to Exogenous Peroxidase. The title summarizes the paper. The peroxidase in the title is a stain, and it's being applied to the organism. It's exogenous, that is, it's not intrinsic to the mouse itself in this case. And they're looking at the localization or the distribution of the stain at the interface between the circulatory system and the central nervous system where the blood-brain barrier must lie. In the abstract, or the summary of the paper, Reese and Karnovski state, horseradish peroxidase was administered to mice by intravenous injection, and its distribution in the central cortex studied with a recently available technique for localizing peroxidase with the electron microscope. So they're using a high-power electron microscope in this study, amongst others. They say brains were fixed by either immersion, dropping the brain into fixative, or by vascular perfusion, 10 to 60 minutes after administering various doses of peroxidase. Like Ehrlich, Reese and Karnovski start with a mouse, and they say that they're injecting a dye, in this case a peroxidase derived from horseradish peroxidase, and they're injecting it intravenously into the mouse. And in order to do that, they need to cut the aorta from the heart so that they relieve some of the pressure. So now as they pump the dye into the circulatory system, they call this a perfusion, so they're pumping this intravenously into the mouse, the dye enters the circulatory system and it replaces the blood that comes out of the cut aorta. And then you have the stain, not only within the blood vessels of the mouse, but the dye has the ability to leak out into the surrounding tissue. I mean, that's what Ehrlich showed. The dye would enter the entire organism with the exception of the brain. Here, the brain. After they perfused the dye into the organism, they needed to fix the tissue, that is, preserve it. So they wanted to fix it by perfusing the fixative into the organism. So they do that. They pump in fixative, and then they prepare the tissue, and they look at the tissue by light and electron microscopy, and they realize there's no dye. And the reason that there's no dye, they talk about this in the results section, is because they pump the dye out with the fixative. I mean, if you pump in the dye and the dye replaces the blood, when you pump in the fixative ended up replacing the dye. So they tried the experiment again, and they perfused the organism with the horseradish peroxidase, the dye or the stain, and then instead of perfusing the organism with the fixative, they dissected out the brain and they dropped it in a vial of fixative. They call this submersion. I'm sorry, actually in the paper they use the term immersion rather than submersion, but immersion and submersion, well, they mean the same thing. Brains were fixed by either immersion or vascular perfusion, is what the paper states. And the vascular perfusion didn't work because it pumped the stain out of the organism, out of the tissue. But it turns out the immersion technique worked. And let's talk about what they saw. To start, they decided to take a look at the brain tissue. It was immersed or submerged in the fixative after stained with HRP by light microscopy. So they take the sample out, the brain out, and they cut it into sections. And then they lay one of the sections on a slide. So looking down from the top, it would look like this. And then if you were looking across, you'd see the slide and then a slice of the brain tissue. And they just put this on a simple light microscope. And in figure 1, they show a photograph of what the tissue looks like. And you can see that you have a photograph like this, and then you have some sort of black stain here. The black that we see is the horseradish peroxidase. It's the stain. And you can see that this is the beginning of the tissue here. So this bright area up on top is just part of the slide where there's no tissue. So you're looking at a piece like this, something like this, and there's areas here where there's no tissue. And then you can see the edge of the brain slice, and then you can see these squiggles that enter down into the sample. And the black lines are the blood vessels filled with horseradish peroxidase, which in this case looks black. And then this area here that's white is the brain, is where the neurons are. So clearly the HRP was able to make it into the blood vessels of the brain, but couldn't make it into the brain tissue itself, into the neurons. It stayed solely in the circulatory system, within the blood vessels. Pretty cool. In order to investigate the blood-brain barrier and figure out what the barrier is, that is, what is keeping the dye within the circulatory system, within the blood vessels, and not allowing the dye to enter the brain, or the central nervous system, they wanted to use an electron microscope because it magnifies at a much higher level than light microscopes do, and they could look at the individual cells and the subcellular structures that might form the barrier itself. So if we talk about the magnification between a light and an electron, so let's talk a little bit about magnification between a light and electron microscopes. Light microscopes, compound light microscopes, have microscope objectives. You all know that. There's, for instance, a 10x or a 20x, 40x. And the highest objectives are 100x microscope objectives. And the light comes through the objective and it bounces off a mirror, and then it goes to an eyepiece that has optical elements in it, and the eyepiece magnifies the subject 10x. So the total magnification of a light microscope is 100x times 10 is, of course, 1,000. So something that's 1 micrometer, or 1 micron, which we all know is 1/1000th of a millimeter, if it's 1 micron in length, then it will be magnified 1,000 times to be 1 millimeter. And if it were 1 millimeter, and we multiplied that by 1,000, then an object that's 1 millimeter in length is magnified to 1,000 millimeters, or 1 meter. And that's the limit of the light microscope based on the wavelength of light, something known as the diffraction barrier. However, in an electron microscope, where you're using electrons rather than photons, the wavelength is much finer than photon wavelengths, and that allows for greater magnification. And in an electron microscope, you can magnify something 200,000 times, which means that if the object were 1 millimeter, that would be 200,000 millimeters, which is 200 meters, which is about the distance of 2 football fields combined. So something that's 1 millimeter in length would be magnified 200,000 times, about 2 football lengths in size. And then in the electron microscope, you're just looking at a very tiny piece of that. So there's a huge amount of magnification in the electron microscope, and it allows you to look at the fine structural details. They say in the paper, fine structural localization of the horseradish peroxidase at the blood-brain barrier. For electron microscopy, we have to take our sample through a few more steps. We have our brain that came from a mouse perfused with horseradish peroxidase, the stain. The brain was dissected out and then immersed or submerged in fixative, which preserves the tissue, it preserves the sample. Then we need to stain the sample with other stains that will stain the membrane, proteins, and other structures, so we can see other aspects of the tissue. And then ultimately it's placed first in ethanol, ETOH, which removes the water, so the ethanol replaces the water in the sample. And then we immerse the sample into plastic, epoxy, like glue. And the plastic replaces the ethanol, and it penetrates the tissue, and then ultimately the epoxy and the sample is placed in an oven, and the epoxy solidifies. So now we have a sample embedded in plastic, similar to a mosquito caught in amber. At that point, we have a block of resin, epoxy, with our sample in it, And you can put this in a machine, a microtome, that cuts very, very thin sections that are 70 nanometers, very thin, 70 nanometers thick. And the sample has to be very small because we place it on a disc, a very small disc that's only 3 millimeters in diameter. This is called a grid. That's the microscope slide for the electron microscope. So we have a section, and we lay the section onto this very small, very thin disc, and then we can put it in the electron microscope to make our observations and photograph our results. In figure 2 of the paper, we can see an image taken with the electron microscope of the interface between the circulatory system and the brain. And so what we see is something like this, and we see these big dark ovals, black ovals. And it turns out that the ovals are red blood cells. And this here is the blood vessel. And outside here is the brain. And you can see that there are other structures that are stained in here. There's circular structures. These are the neurons and astrocytes and so on of the brain tissue. And in the lumen, the inside of the blood vessel, you can also see this very granular stuff in here. And the granular stuff is the horseradish peroxidase. And the point of this figure is that you can see the HRP filling the lumen of the blood vessel and surrounding the red blood cells, but it doesn't get into the central nervous system. The red blood cells are black because they have their own endogenous peroxide. It turns out when these experiments are done after the HRP is administered, the samples are subjected to some other chemicals that turn the HRP black, as well as the endogenous red blood cell peroxide. So the red blood cells turn black, the HRP turns black. The cell membranes and subcellular structures that we can see in the central nervous system were stained with the additional stains we added in processing the sample for electron microscopy. At higher magnifications in the electron microscope, as shown in figures 3, 4, 5, and others, you can see the endothelial cells. These are the cells that line the circulatory system, and I'm going to try to draw this. So here's one endothelial cell, and another endothelial cell, and a third, like this, and then you have red blood cells in the lumen. This is the lumen of this here is the lumen, the inside of the blood vessel. Another red blood cell, and so on. And as we saw before, the HRP is extremely granular, and it's found throughout the lumen of the blood vessel. And you can see that some of the HRP comes up here, but at some point the two endothelial cells are so tightly opposed to one another that they're linked here, and the HRP can't get through. It stops, and that's found in a few different examples in the figures like this. It comes up and then it stops because these, I didn't draw it very well, but these are very close to one another, probably held together. Well, now we know they're held together by tight junctions. So these cells are very tightly held together, and the HRP can't get through, and that's the blood-brain barrier. So figures 4 and 5 show a close-up of these interactions between endothelial cells and show that the HRP can't get through the tight junctions that are holding together the endothelial cells. The endothelial cells form a sheet and they inhibit the HRP and other substances, including many medications, from getting from the circulatory system into the central nervous system, the brain. The last figure I'd like to talk about is figure 8. During their experiments, they noticed that in some cases you could see endothelial cells that had vesicles inside the cell that were filled with HRP. So if we redraw the blood vessel, here's another endothelial cell and another. Like this, and this is the lumen of the blood vessel where we have our red blood cells and, more importantly, HRP. They could see that in some cases, if I redraw this cell bigger, so I'm going to draw it like this, they could see these endocytotic processes happening where the HRP, this is this membrane here, that the HRP could get into this during this endocytotic process, and then this pinches off to form a vesicle. They say it's a pinocytotic process. But this is interesting because it means that the HRP from the lumen can get into a vesicle through this endocytotic process, pinch off, and form an HRP-filled vesicle. And then this vesicle could fuse with this membrane and then release its contents into the brain, because this is the brain out here, right? Brain in brain. So this suggests that there's a natural process where some of the material from the lumen of the blood vessel in the circulatory system actually could get across the blood-brain barrier. Okay, that's it for Reese and Karnovski. I wanted to mention that Tom was my postdoc advisor at the National Institutes of Health, and we worked for many years at the Marine Biological Laboratory in Woods Hole, and Tom's one of my very closest friends. I also knew Morris Karnovski. He died a few years ago, but Tom and Morris did this work at Harvard Medical School. The paper that we're going to read on Friday was by Milton Brightman, another friend of mine, and Tom Reese, and they look at the second part of this very interesting story. Thanks, everyone. See you next time.